So hi everybody, my name is Jessica Rohde and I'm here to introduce our next two speakers. Neelu and Sara are here because of IARPIC's new effort to engage early career scientists. And you might have also noticed that at IARPIC we've been putting more of an emphasis on having shorter presentations, especially what we are calling lightning talks. And so Neelu and Sara and 6 other early career scientists have spent the last few months taking an online course from me that teaches the use of effective interdisciplinary science communication. And so these talks that you're going to see today from them are their final projects, and they'll be presenting them to you today as well as an IARPIC-wide public webinar next month. So please welcome Sara Pedro, who is a PhD student at the University of Connecticut, and Neelu Singh, who is a PhD student at Mangalore University and National Center for Antarctic and Ocean Research. They're both going to speak about different aspects of marine contaminants. And these presentations are short, just 5 minutes, so I hope you'll stick around. They've worked really hard on them and are really happy to be here. And they're just designed to introduce you to their research so that they can start building connections and collaborations through IARPIC. So if you're interested to know more, please follow up with them, and I will put a link to their IARPIC profiles in the chat box. So Sara, if you want to go first, you can start sharing your screen.
Hi, everybody. All right, let me just do this. Okay, can everybody see my screen and hear me well?
Yes.
Great. My project has been focusing on how marine species moving up to the Arctic may alter the way contaminants are transferred in the food web. And I basically have 2 projects. One focuses on fish in the Canadian Arctic, but today I'm going to talk about the other one that focuses on killer whales moving up to Greenland. So the sightings of killer whales in the Arctic have increased exponentially since the 1800s to 2000s, particularly in very recent years. And these have occurred in the Canadian Arctic, but also in Greenland, and in the Chukchi Sea, there's also some evidence as well. So they might be expanding their habitat range into the Arctic because there's less ice now, and when they come up, they're generally observed feeding on Arctic marine mammals like beluga whales or bowhead whale. And what we think is that in the North Atlantic waters, we probably see more of, this scenario. So the left side where the killer whale feeds on fish. And in the Arctic, we might see more on the right side scenario, killer whales feeding on marine mammals. And we are concerned about this because they might be increasing their exposure to contaminants that accumulate in the food web. So marine mammals have higher levels of these contaminants compared to fish. And some of these contaminants are polychlorinated biphenyls and organochlorine pesticides, which are basically man-made, and they get to the Arctic, and they are of concern in Arctic food webs. So we got some samples from Southeast Greenland that were from harvest from the indigenous communities. And there are about 16 killer whales of different sex age classes. And some of them had marine mammal remains in their stomachs, in fact. And we also have a couple of stranded whales from the faraway islands that we are kind of using as a control because we know they were in North Atlantic waters. So overall, we see variation in the contaminant levels in different sex age classes. So you see different colors correspond to different classes, and the highest levels were generally in sub-adults, and the one adult male collected compared to adult females, which is probably related to the reproduction because they can release some contaminants to their offspring. But this variation didn't quite explain why in Southeast Greenland the levels were much higher, so on this graph, compared to in the faraway islands. So we think this might be due to diet, and in the faraway islands, the killer whales might be eating fish, whereas these ones in Southeast Greenland had marine mammal remains in their stomach, so we know they're eating marine mammals. Another point is that the levels of these contaminants were quite high in the killer whales in Greenland, so above threshold levels for toxic effects that were observed in other marine mammals in the wild. And our results kind of go along with what is observed in other populations of killer whales. So in the Northeast Pacific, there's two different ecotypes that are classified based on what they eat. So the transient killer whales eat mainly marine mammals, whereas the resident eat mainly fish. So we see this huge difference in their contaminant concentrations, about 10 times higher in the ones that eat marine mammals. And then when we look at the North Atlantic, so in Norway, there's a killer whale population that mainly feeds on fish. And compared to our results, it's about 2 to 3 times lower. So what we think is that in these killer whales that are coming to the Arctic still eat fish, but they are eating marine mammals more consistently, and probably it's a seasonal habit when they come up to the Arctic. So their contaminant levels are higher. In terms of future directions, and again, those those levels of contaminants were so high that we're curious to see if there's killer whales are going through any kind of toxic effects from these contaminants. So what I'm doing now is trying to correlate the concentrations of contaminants with biomarkers of toxic effects. And I'm using vitamins A and E as biomarkers to try to figure out if they are suffering health effects from these toxic levels, from these high levels. We don't know if they're toxic. Another thing to think about is that in Greenland, at least, they are eating these killer whales, and because they have such high contaminant levels, we don't know exactly if people will be exposed to any significant levels as well. So it would be something to consider in the future and try to look up as this new predator is coming up to the Arctic and being available as food for people as well. And that's all, thank you. If you have any questions, I'll be very happy to answer.
Are there any questions for Sara?
Do you have any data on body burdens for fish in the region?
Could you repeat?
Do you have data on body burdens of organochlorines from fish from the same region where the whales are from?
We do not, but what we also doing, and it's coming out, is other people in my lab, is measuring fatty acid levels. So we can track what they're eating based on their fatty acid signatures. So it's kind of a dietary tracer. And through that, we could tell that these killer whales were feeding on marine mammals and also fish. But in terms of the contaminants, we don't really have the concentrations for fish in this region, but it's basically based on what the levels generally are for marine mammals, because they are above in the trophic position, they tend to have these higher contaminants. I don't know if I answered your question right well.
Sara, do we know anything about the same type of research on the Pacific sector of the Arctic?
I think there was one study, I haven't looked at it recently, but there's some evidence that they are becoming more abundant in Alaskan regions as well.
Okay.
There is some work, I don't know about these kinds of toxins, but there are some people looking at HABs, harmful algal bloom, saxitoxins, in intestines of walrus and other organisms, because they're obviously that's what they feed on up here. So there's an increasing interest in getting some background information on that, because as we know, these HABs exist everywhere. But though they may not be at levels that are a problem, so they just want to get some background information to see what the levels are. And I think so far they've been really low. But I also know that there's some work going on looking at tissue from whale landings, from the native bowhead whale landings and whatnot. So there is some work going on up here, but I'm not sure if there's anyone doing this for orca specific whales in looking at toxic contaminants like this.
Thank you. Any other questions? If not, we can switch to the next speaker. Jessica, you want to say anything or to introduce Neelu?
So Jessica had to jump off. So I'm going to share Neelu's profile on the IARPIC website, and Neelu, you can start whenever you're ready.
Hello, I hope I'm audible.
Yeah, it's better.
May I start now?
Yep.
Yes, please.
I hope my screen is visible to everybody.
Yes.
Okay, so first I would like to introduce myself. I'm Neelu Singh, and I'm pursuing my PhD from NCAOR, National Center for Antarctic Ocean Research, which is located in Goa, India. And my topic of today's talk is spatial distribution of heavy metals in the surface sediment of Kongsfjorden, Svalbard. So first, I would like to talk about what heavy metal is. Heavy metals have many definitions, but from environmental perspective, any elements that has a high density as compared to water and associated with contamination of potential toxicity is termed as heavy metals. And these heavy metals can enter into the environment from the natural sources like volcanic eruptions, forest fire, as well as from the anthropogenic sources like burning fossil fuels, industrial waste, municipal waste burning, etc. So why is it important to study heavy metals? It's important to study heavy metals because these metals, they can become persistent environmental contaminants because they cannot be degraded or destroyed. So, along with that, they have a negative impact on the human and ecosystem when their concentration is above from their background values. How these, what are the modes of them that they reach to the Arctic? So mainly they have two modes, from the air currents and through the ocean currents. They reach to the Arctic, and along with this, on the local scale, they can enter into the environment through the glacial meltwater as well as through the terrestrial sources. Now I would like to tell you about few things, my study area. As I said that it's Kongsfjorden, Svalbard. Here on the my left hand side, you can see that there is a map of Spitsbergen, Svalbard, and there is a red circle, oval circle. This is the location of Kongsfjorden. And on the right hand side, you can see that star. This is the location marked from where I have collected my samples. And this Kongsfjorden, it's considered as a unique night because this Kongsfjorden, it occupies a unique night in the Arctic climate system because it is maintaining a delicate balance between the advection of the warm saline Atlantic water at its mouth. Here you can get one number. It's a mouth, and 9. It is a glacial side. So it is like at its mouth side, and that from other side, that station number 9, there is a glacial runoff from the large tidal glacier. I started my studies with two objectives. The first one is like I want to contribute to the database of contaminants and Neelu, because when I did my literature survey, then I come to know that there are many studies on the trace metals and heavy metals in other sectors like air and the zooplanktons and other. But in sediments there are very less scanty studies. So I thought that my study should help to contribute in the database of contaminants. And another one is to investigate the trends in the variability of heavy metal from their anthropogenic influences. Like we know that in that Neelu area nowadays there is a growing human activities, and that it is becoming a tourist hub nowadays. So it's like to understand whether these activities are affecting the concentration of heavy metals there or not. So from my observation and my analysis, I come to know that that the concentration of heavy metal, when I compare to the other pristine regions in the Svalbard, it is comparable, or even some cases it was less to this another locations. And when I calculated the enrichment factor is like when you calculated it, you can come to know that your data are coming into which class. It's like it have 3 to 4 class. It's like not enriched, slightly enriched, and the polluted. So my analysis, that my data, it comes under the not and the slightly enriched. So we can say that for now, this heavy metal concentration is not like it's not influenced by the anthropogenic activities. So we can say that it is a negligible. But I just want to make a point that okay, today it is not too much, but as I said that these metals, they can persist and it can become they can bioaccumulate and biomagnify. Maybe today it is less, but maybe we can expect them with the time they can increase in their concentration, and they can reach up to the peak levels. Thank you.